So I am very happy to be able to talk about this. This is one of the most exciting areas right now in multiple myeloma. I would like to start off by just saying I'm not a bench researcher, immunologist, but I also trust that not very many of you are either. So we're going to sort of muddle through this, and what my job is really today is to sort of translate some of these emerging basic science discoveries and how they're coming into the clinic right now in immunology and how we're applying them to multiple myeloma. So we saw a table similar to this actually in our last talk and others, where we think about agents in myeloma therapy according to the class, right? We're used to the idea of, we've heard about IMiDs, we've heard about proteasome inhibitors and other specific things. We've also heard about monoclonal antibodies already, right? There are 2 FDA-approved monoclonal antibodies in multiple myeloma. Daratumumab, which targets CD38, and elotuzumab, which targets SLAMF7. Those are forms of immunotherapy. So it is already here, but as you can see, looking further down this last column, there are a lot more coming. And actually, within immunotherapies, there are several different types of immunotherapy. So unlike some of the other columns where there's really kind of one class of drugs and a bunch of drugs that are within that one class, these are all very, very different types of therapy in the immunotherapy column. And I'm going to try and sort of walk us through them. The one part I'm not going to get into today is checkpoint inhibitors. It's just because we have a lot of other things to talk about, and I know specifically CAR T-cell therapy is something we all want to hear about. And so we're just in the, because of time, we're not going to spend a lot of time talking about that one topic. So just a little bit of background on immunology and immune theory and why we're interested in this. The immune system is your defense mechanism against the things that are out there in the environment, bacteria, viruses, fungi, parasites. It's also the part of our body that gives us seasonal allergies and other good things like that. But basically, it's how our body reacts and keeps us safe from things in the environment. And there are different types of immunity. There's innate or natural immunity, and then there's adaptive or acquired or specific immunity. So natural immunity are things in your body that are always ready to defend you, and it's your first line of defense against invaders that get into your body. So, for instance, your skin. Right? I mean, we have barriers. We have natural barriers that are part of, that help keep us safe from the environment. We make antibodies. That's a specific type of immunity. However, over time, we're exposed to many, many, many things in the environment. And so we sort of have many of us have sort of a resting cadre of immune cells in our body that are sort of ready and able to take care of the things that we come in contact with the environment. Similarly, other types of adaptive or acquired immunity would be things like vaccinations that we're going to talk about. Vaccinations work by the same mechanism. As I was just mentioning before, you're exposed to something, and something is administered, usually as a shot, some vaccines as intranasal inhalation, but you're exposing your body purposely to something to try and generate an immune response. And once having generated that response, your body has memory. Your immune system has memory, and you are able to respond again in the future more quickly and more vigorously once you've had that initial exposure. That's the basis of vaccines. So the question is, how is the immune system relevant to myeloma? Right? If the immune system's job is to find things that don't belong in your body and fight them, why don't myeloma cells, or for that matter, other cancer cells, come under attack by the immune system? One possibility is that they just look too much like normal cells, so they're not identified as foreign. Right? These are cells these are cells that are derived from an original cell that was in your body. right? So many of the features of the cell are similar to the cells, other cells in your body. Also, myeloma cells the markers or proteins on the surface of myeloma cells may be presented in a way that the body develops tolerance to the presence of these cells. We remember that there's a long pre-malignant state for many patients, monoclonal gammopathy. Myeloma may also take a more active role, may take more active measures to evade the immune system. They may inactivate normal T cells. They may increase the presence of immune-inhibiting cells in the bone marrow microenvironment around them, they may also stimulate other pathways within your body to just downregulate your immunity in general. So there are many, based on all these different potential mechanisms of inhibited immunity, there are different ways perhaps to apply immunotherapies to look at this. So, for example, we talked about the approved ones, monoclonal antibodies, right? Monoclonal antibodies are a form of what we call a passive immunotherapy. We're not asking your body to do anything much. We are providing an antibody that targets your myeloma cells. Another non-specific form of immune therapy is IVIG, right? We're giving you antibodies against things that are in the environment, pooled donor immunoglobulin, to prevent infections. That's non-specific because it's not targeting any one specific thing. It's targeting a lot of different things. But monoclonal antibodies are another form of passive but specific immunotherapy. And we're going to talk about some other new emerging monoclonal antibodies. We also can do cellular therapies, and the one we're going to focus on really is CAR T cells. CAR T cells are modified immune cells that they're actually cells taken from your body, modified modified in a way where they will fight myeloma better, to overcome the roadblocks that are present in your system prior to that. And then we have IMiDs, checkpoint inhibitors, other medications that overcome immune suppression. And then I mentioned before we have vaccines. And although vaccine therapy is probably bringing up the rear in terms of progress overall among these different choices, vaccines may actually have the most promise in some respects. And there's a ton of work, early clinical studies and bench research being done in the area of vaccines, and we will touch on that a little bit later. So let's start with monoclonal antibodies. There are different types of antibodies. Now, Craig showed you a schematic of an antibody before. He pointed out the heavy chains and the light chains. And it's important, so on our naked antibody on the far, I guess your left, the blue end there, the blue ends, that's the business end. Think about the antibody like a barbecue fork, and then you understand what the business end of the antibody is, right? The difference, though, is that the end isn't really just like a barbecue fork. It's more like a key. Okay, so you can determine what this antibody will sort of poke at or bind to by programming that part of the antibody. Okay, so that's how an antibody is targeted. Okay, so there are different types of monoclonal antibodies. So again, on the far left, there's a naked antibody, which is just an antibody that coats myeloma cells or some other target and may damage the cell directly or may help your immune system find those cells better. A conjugated antibody is actually a form of targeted delivery. You can see here that attached to the tail end of this antibody is a toxin, or you can also attach a radioactive isotope. And when you do that, what you're taking advantage of the targeting on the other side of the antibody, and you're delivering something that's poisonous to the myeloma cell on the back end. And when an antibody binds and is internalized in the myeloma cell, it kills it. And then Dr. Chari mentioned bispecific antibodies, and what you can see here in this schematic is that the antibody binds more than one target. You can see that there's a pink side and a blue side, and so those 2 points, okay, those 2 tines on the barbecue fork don't target the same thing. And there are reasons, I will show you why you might want to do that, what you could do using that strategy. But also there are different constructs that can serve similar purposes, and BiTEs are shown here. And basically what you can see schematically is this is the sort of the business end of the bispecific antibody without the actual antibody attached to it. It's just like with a linker protein rather than the rest of the antibody. So as an example of a naked antibody that's in development in multiple myeloma, we have isatuximab. This is a naked anti-CD38 monoclonal antibody. CD38 is the same target that daratumumab targets. And isatuximab is far along in development. There have been many studies done with this antibody. It targets CD38 in a slightly different way than daratumumab. And you can see that it is being developed as a single agent therapy. It's also being studied in combination with other agents, as shown here. IMiDs, proteasome inhibitors, and others. And you can see in the far right column that patients can respond to these agents, to isatuximab, even after several lines of therapy, 4 to 5 lines of therapy. Importantly, for most of these trials, though, these trials are not generally in patients who've had previous daratumumab. And that's an important question that is actually being explored right now by the makers of this antibody, is if you have a patient who stops responding to that particular CD38 antibody, can you target it with a different CD38 antibody later? We will find out. We will find out the answer to that question. But at this point, we don't know. As an example of a different type of antibody, a conjugated antibody, this is the one that has the payload attached to the tail end. And this is a diagram of an anti-BCMA antibody. BCMA is B-cell maturation antigen. That's the target on the myeloma cell, on the plasma cell. And the toxin is auristatin F, which is a toxin that has been used in many different conjugated antibodies that have been designed to target many different proteins in many different diseases. But this is an exciting one, and that has gotten a lot of attention recently in myeloma. This is just a phase 2 trial that involved 35 patients, so 36 patients. But these patients had on average 5 prior lines of therapy, and this 1-hour infusion of antibody every 3 weeks led to a 60% response rate. So that's different than the 20 to 30% response rate we were hearing previously. So this is, and this is, you know, there are other examples of drugs that have been developed that had this kind of activity as a single agent in refractory patients. And what we know is that response rates like this in heavily pretreated disease is really important. So this is something. There was some toxicity, however. There were eye problems seen, and it is not 100% clear whether or not that was due to the target or to the toxin. I suspect personally that it was due to the toxin, as other antibodies that target other proteins that have auristatin F in it also had some corneal toxicity. This is manageable, but does require eye exams and eye screening if you're going to use this. And also there was some effect on platelet counts. So this is exciting, an anti-BCMA monoclonal antibody conjugated to auristatin F. So let's go back briefly before we move on from monoclonal antibodies. Let's talk about the bispecific antibodies, and in the next few slides, we're going to abbreviate it as bispecific antibodies, BSAB. And they do have multiple potential uses. So this is a generic tumor cell. I tried to make it look a little like a plasma cell with different surface markers on it. And one use that Dr. Chari did allude to is the idea of using these antibodies, or you can easily picture the bite that I showed you before, serving the same purpose as a sort of linker to bring immune cells into close proximity to tumor cells, right? So if you have one arm of the antibody that targets a marker on T cells, let's say, and the other arm targets tumor cells, you can actually you can bring these cells together using the antibody. That may be more accessible and easier to apply across many patients than something like CAR T cells for reasons that I'm going to show you when we get to the CAR T cell section. You can also use these kinds of antibodies to actually block receptors on cells. So if you wanted to try and prevent things from binding to other receptors on myeloma cells, you can use antibodies, bispecific or monospecific antibodies. You can use them to bind and block receptors so that they can't function normally. So there are several ongoing studies involving bispecific antibodies and BiTEs. I highlighted the first one not because it's more important than the other ones, but just to show you that this is the example I'm showing you a diagram of, and just the terminology. So when you're reading about a clinical trial and it says CD38 by CD3, what that means is it's an antibody that has one arm or has receptors for both CD38, like Darzalex, or isatuximab, and then another one that binds CD3, which is on T cells. It's too early to really discuss the data about bispecific antibodies, but this is something that's being actively explored. The thing about this, now I mentioned that this might be easier in some respects than CAR T cells. One of the things about antibody therapy in general is that antibodies have a finite half-life in your blood. So just like any sort of drug therapy, you need to repeatedly dose antibodies for them to continue to be effective, right? They're not going to last in your system forever. And in contrast, cellular therapy may be different than that, or is different than that. So immune cell therapy, as the slide says, the gift that just keeps giving. And what I'm referring to in this slide is the idea that you're giving a living therapy. You're giving, we're using myeloma-directed immune cells, in the case of CAR T cells, T lymphocytes, and we're modifying the cells in a way to fight the cancer cells specifically. We extend them, meaning we collect cells from the patient, we modify the cells, we expand them to create the cell dose that we need to effectively target the myeloma, and then we reinfuse them, and hopefully they engraft. Right? They're living cells, and hopefully what you end up with is a standing, living, thriving population of cells in your body that will continue to fight the myeloma. That's the promise and the attractiveness of this type of therapy. There are different types of immune cell therapy. We're going to focus on CAR T cells. There's a similar type of therapy called T cell receptor engineered cells. And then there's another type of therapy called marrow-infiltrating lymphocytes. All I'm going to say about those is what we know is that in the bone marrows of patients, there are populations of lymphocytes already in the bone marrow that have anti-cancer activity. And those cells are present in varying degrees in different patients. And there is a lot of interest in trying to identify those cells, extract them, expand them, and then reinfuse them as a form of therapy. So that's what marrow-infiltrating lymphocytes are. So normal T cells versus CAR T cells. So the analogy here is that you have a natural, normal, non-modified T cell with normal T cell receptors, which may be able to fight myeloma cells or other types of cells, but they need to be stimulated to do so. They're not inherent; they weren't born to fight myeloma cells specifically. They might be goaded into it. CAR T cells, on the other hand, are engineered to specifically home in on myeloma cells by modifying the receptors that are on the surface of the cells. And I think you can probably, with your imagination, see how those receptors on the surface of the cells look a little bit like antibodies, right? The structure of the antibody that I showed you. And so those receptors are modified in a way to make them very specific for myeloma cells. So briefly, what do we do? Why we've all said that CAR T cells are not super easy to give and to deliver. Why is that? Let's talk about what it actually involves. The first thing that we need to do is we need to do leukapheresis. Leukapheresis is similar to what we do with stem cell collection. It takes place in a pheresis unit. And what we're doing is we're skimming out different cells than the stem cells. We're skimming out white blood cells and isolating and stimulating a certain subset of T cells. And then we do a transfection procedure where we introduce, where we modify the genes of that cell population so that the T cell receptors so that the receptors on the surface of the cells are modified. And then once we have those genetically modified cells, again, we expand them to achieve a certain cell dose. And then we reintroduce them as an intravenous infusion, as I mentioned before. That's when they can go to town and start trying to attack the tumor cells. There are numerous CAR T cells in development. There have been CAR T cells developed against different markers such as CD19, CD38. There has been interest in SLAMF7. But the most work right now is being done in that same target that we saw with the conjugated antibody before, targeting BCMA. And this is a table that shows 5 different studies looking at BCMA-targeted CAR T cells. And what we see here, again, are very high response rates in patients that have had a lot of prior therapy. The response rates range from 50 to 100%. This work is being done all over the world. China has actually been very active, but there are many centers in the United States that have been working on this for quite some time. There are complete responses seen, importantly, in heavily pretreated patients. There are some real toxicity issues that need to be sort of worked out. The most important of which is cytokine release syndrome, or CRS, and this is like a sort of a systemic inflammatory response. The best, I guess, analogy I can think of is it's almost like a septic or sepsis type picture where you can have problems with fluid shifts and blood pressure and fever and breathing, and it is an inpatient requiring risk. CAR T cell therapy is not given on an outpatient basis. There is a sizable risk of this. You can see that the majority of patients using CAR T cells as they are currently formulated experience these kind of reactions, and up to a third of these patients, these are very severe reactions that land patients in the ICU. We also see some neurotoxicity with in some of the studies as well, and so that's also something else that needs to be worked out. So what I think you can see with several different centers working on CAR Ts, and the last table was just the ones directed at BCMA, that we have a race amongst different companies to get these going, and right now several companies are rapidly expanding their clinical research efforts with this. We need to improve the efficacy. We need to figure out why 100% of patients on all the studies aren't responding. We need to understand why CAR T cells may work for a period of time and then stop working. And we need to figure out how to, with next generation cells, figure out how to make the cell populations more durable within your body once they've been infused. We need to improve the safety with the cytokine release and neurotoxicity that I mentioned. One potential thing, if later toxicities are noted, is the idea of introducing a safety switch. That is, when you're genetically modifying the cell, introduce something that would allow you to target the cell with a common medication. And so, for instance, make it susceptible to acyclovir, make it susceptible to some other medication that you could just give somebody, and that way you could turn off the cells if necessary. And then we need to improve access. Right now, you need to be able to go to a center, have your cells collected through pheresis, have that center be able to modify them and then give them back to you and then handle all the toxicities that go with it. That's not therapy for everybody right now. That's not even to mention the fact that there are some people who just aren't fit enough to handle the cytokine release, right? Improving access by creating what we call off-the-shelf CAR T cells, that is, cells that aren't your cells, but cells that are just stored, cells that you could just order from a pharmacy, and thaw and administer, and they don't have to be your cells. That would be a major benefit because it would save several of the steps that we're talking about already. And then, of course, we have to figure out what the best targets are for CAR T cells. We're focused on BCMA, but there might be better targets developed, and we will see. Cancer vaccines, just like monoclonal antibodies, don't all mean one same thing. Cancer vaccines don't either. Again, I talked to you about the strategy or idea of vaccines before in general. Cancer vaccines can be cell-based, where you administer a cell that presents some protein or other antigen that your immune system responds to. You can also actually give sort of like ground up or lysed tumor cells, and then all the things that are in that sort of tumor cell soup. If you administer like that could be used to stimulate an immune response. And then there are non-cellular therapies, vaccines, where you just administer some sort of protein or molecule that stimulates an immune response that will be useful because there'll be cross-reactivity with tumor cells. To prevent vaccines, as we use them to prevent infections, are given ahead of time as a preventative. Cancer vaccines are generally being administered as a therapy for an active disease. The one exception in myeloma might be if we were trying to do a vaccine strategy in a patient with smoldering myeloma in an effort to prevent it from becoming full-fledged myeloma. Currently, most of the vaccine trials that are being done administer them in sort of the post-transplant or post-induction stage when the patient's myeloma is at its most reduced, and also before they've had line after line after line after line of therapy, over which time their immune system may become a bit less robust. So the perfect sweet spot may be fairly early in the course of disease, but when the disease has been reduced significantly and it's on its heels and it's at its most vulnerable point. And I'm not, obviously we don't have time to go through these tables. I'm showing you this slide to show you that there are multiple multiple labs and clinics working on cell-based myeloma vaccines, and then non-cell-based vaccines as well. So just between these two slides, I'm showing you about 10 or 12 different vaccine strategies that are being explored right now. So we're looking forward to having these. If a vaccine can be developed, it would be extremely useful in selectively targeting malignant cells, maybe useful in targeting different tumor clones, and has the advantage, like cellular therapy, of being a long-term therapy that can provide benefit to patients without having to give repeated doses of a medication.
I really thought that was the best talk that I've seen on this subject. And I think it's complicated stuff, and it's new stuff. We have one question. Are there any CAR T cell clinical trials taking place in the state of Michigan?
Not on this exact date, but I believe both of us have been working very hard to make sure that CAR T cell therapy will be coming to Michigan. I don't think we can promise a date, but I think...
Oh, I mean, I think we're about 4 months away.
I think, exactly. I think both centers are close to having CAR T cells. So I think we can anticipate having access to that in the state of Michigan in this calendar year.
Perfect.
Yep.
Thank you, Dr. Zander.